I'm Maria Wilson.
And I'm Danielle Mandikian.
And we are scientists. We love science.
Yeah, we do. So when we aren't doing it, the next best thing is to talk about science. And what's really awesome is that we're surrounded by some of the most brilliant minds in research.
We're going to step away from the labs today to talk to other scientists about the cool stuff they are thinking about, working on, and imagining.
As well as how some of these discoveries just might lead to new medicines. So grab your favorite drink, get ready to unlock your science brain, and join us for Two Scientists Walk Into a Bar.
The show for scientists, science geeks, and the people who love them.
Can you define an organoid?
It's sort of like a in vitro organ mimic.
It's like a robot in organ.
A what? An organoid.
An organoid. Yeah. Yes, I can define an organoid. It's where you take tissues, like primary tissues, and you're able to grow them in a more similar structure to the primary organ, but in a dish or a more microscopic scale, and then use that for experiments to better recapitulate the actual organ environment.
Hi, everyone. Welcome to today's episode where we're going to actually dive into the coolest topic that everybody who's in the know is talking about, organoids. And we have with us our special guest, Kim Homan. Welcome to the bar.
Thanks for having me.
What are organoids? What's all of the buzz about?
Organoids are a way to model human organs in a dish, right? So organoids, I guess, can be defined as starting from stem cells, which can then derive other cells in your body. You usually put them in some sort of 3D ball in a dish, and through coordinated timing steps of giving them certain additives in their media or the thing that feeds them and houses them, you can direct them towards certain lineages of the body. So you can make a little mini kidney organoid or a little mini brain organoid. And we've even gotten sophisticated enough to do different brain regions.
Really?
Yeah, like dorsal forebrain and hindbrain and midbrain and things like that. So these are not perfect mimetics of the organs, right? So I want to be very clear about that. But they start to have the diversity of cell types, and it gets really interesting to study them because you can also genetically engineer them, and then people with certain, for instance, mutations, you can start studying those mutations in a dish and how they actually end up as a certain phenotype.
So let's take that definition a little bit further. So for all these different organoids, like how big do they have to be? How long are they, what's the shelf life? Do we have to feed them? What's another way that we can kind of classify them?
So they vary in size. I would say anywhere from, couple, like 100 microns, which is, I guess, half the width of a human hair, all the way to millimeter-sized constructs that you can see physically with your eyes. Certain ones, for instance, like brain organoids, they actually grow in size the whole time. They grow fairly normally. You do feed them. You have to give them, nutrients and remove their waste, just like that would happen in the body. Once you get to a certain state with the organoids, typically there's kind of like a maintenance period where you can kind of just keep growing them for a really long time, typically. So, for instance, researchers, one interesting tidbit on the brain ones, researchers have gone out past, actually years now. So
Wow.
Same ones for years. And interestingly, at 9 months, there's like a switch transcriptionally. You can see that they went from like a pre to postnatal state, even in a dish.
That is so bizarre.
Yeah. So these cells know what they're doing. Like, it's so weird.
Like, how do you keep them from getting necrotic?
So, okay, that does happen. So if they keep growing like in the brain, and this has been like one of these things that's been debated in the field for a long time, but I feel like it's pretty well known. People know that the center of these things aren't receiving the same, signaling, and there is some metabolic stress. And that metabolic stress actually does show up in their transcriptional signatures if you were to like break them down for single cell sequencing, for instance. There's other organoids that stay smaller, and they maintain a certain amount of diffusion that can get to the core, and then they don't have as many of those problems. So each organoid type is kind of its own baby, and they can make them over, I've definitely seen, 10s, 20 of these types already, if not more, that I'm not even mentioning.
Two-part question. First part, what are the kinds of questions you can use organoids to answer? High level.
High level. So you can use organoids to ask about toxicity. You can use organoids to model disease and then try to understand the specific pathways for which that disease evolves, right, and the phenotypes that evolve out of that. You can ask them about disposition across a barrier, for instance, if you integrate them, especially in an organ-on-chip system, for instance. And you can ask about biomarkers and development of biomarkers to look at either a disease or an efficacy of a specific treatment.
That leads me to my second question.
Okay.
So at what stage in the development process do we use this?
Very early to very late. So across the whole spectrum, they can be applied. So if you look at the early stage, when you're just trying to understand a disease, right? And then you start thinking about all the ways to model that disease. Like, oh, do I go to an animal model of that disease, and is that animal model predictive? Or can I actually go to a human organoid system, make some genetic modifications, or make some physical modifications to give it a disease-like state? And then can I try, different pathways to modulate, to ameliorate that state, right? So that's super early. You can use it as early, as just those thoughts are happening. And then throughout development, right, where we ask questions about disposition of the compound across different barriers and what is the drug on healthy organs, like that you weren't intending to hit. Then for planning clinical trials, like you can look at these things for trying to plan, combination trials. Are if I put these two drugs together, would it be like synergistic toxicity, or would it be, actually not synergistic, so I can dose both of those pretty high? And then finally, when you're in clinical stages and you get responses you did not a priori predict, you can go back and use these organoid and organ-on-chips to understand why. Like, what is the mechanistic effect that happened from that drug on this particular population? Now I can really look in depth because most of the cell types are there and all of those interactions are possible. And usually you can even visualize the drug with a little fluorophore light attached to it, as you very well know. And study that, right, in a dish even.
So I really want to give you a chance to brag on these because I really think this is one of the coolest areas in research. So when you think about these kind of more complex groups of cells, I don't know quite how to ask this, but what are some of the specific aspects that you really think set these apart for study systems? Is it the 3D nature? Is it having that mixture of cells? What about this is so much more advanced than what we might think of as traditional in vitro systems?
So for traditional in vitro systems, it's usually isolation of one cell type. A lot of times that cell type is immortalized, so it might come from cancer, and then you might grow it on a dish, in a plastic dish in 2 dimensions. And that would be what we've done for greater than 50 years, across the whole industry, right? So that would represent one cell type that's not even really representative of that cell type because it's immortalized. So you took some steps to actually make it grow indefinitely, which is not what your body normally does, right? All the cells in your body eventually die. Turn over at least. Yeah. And so now the cool thing about this is the compendium of cell types that you can get from starting from a stem cell actually replicate. So for instance, in the kidney, there's about 23 known cell types, and the kidney organoids represent almost all of those, in one place, in one little ball. And so the difference is I could take one, proximal tubule cell from the kidney, that's one of the parts of your kidney, one tubule, and I could represent just that, which is what we've been doing for many years. Or then in a 3D setting, in a coordinated way, these cells will actually make all of those compendium of cell types and then give you coordinated responses, right? That's the cool thing that we just couldn't do before.
Yeah, and you think about, too, from a kind of a development standpoint, some of the questions we want to ask, is a therapy going to be useful or whatever? I feel like just having one cell type as a response never cuts it because it's always this like nice coordinated like orchestration of responses or
Right.
whatever the right phrase is, you know.
Or like regeneration, like we want to know if this molecule would regenerate, your gut epithelium, and it's like, well, you have the stem cells there, they will produce all the differentiated types, so now you can try that drug in a dish and you can see if it has that response without ever touching an animal yet, and just dealing in human biology and not in animal biology.
Hi, Danielle.
Hey, Karen.
I'm not really familiar with what a proximal tubule cell is. I can't even say it. What is it?
I mean, who is? I only have like one friend who's like deep into that. So proximal tubule cell, scientists are not creative. A proximal tubule cell is a cell that makes up a proximal tubule in a kidney. So the real question is, what's up with that? Why do we care about proximal tubules? And so it's just, really what it kind of like hails to is that this is a very specific part of this organ that has a really critical function for reabsorption and filtering out certain things that are in circulation. And so if we can try to make that an organoid, we can figure out what's happening to our therapeutics whenever they're going through the body and being processed.
Hey, Danielle.
Hey, Wellington.
Another definition for you. Gut epithelium. I should know what that is, and I feel like if it was on a test, I would probably answer, but it might be wrong.
Well, hopefully a previous professor doesn't hear me give this explanation, but like epithelia, endothelia, you always hear people talk about these different cell types. There's two ways to look at it. First is the very simple answer. What it means is a type of cell that makes up kind of a layer. Right? The cool answer is that what distinguishes them actually happens in the developmental phase when you're an embryo, and it all kind of stems from what type of tissue those originate from. So endothelial, epithelial, they're basically these like cellular layers that occur, and these kind of give us the walls of our organs, and the origin is where the name comes from. So there's so much potential with this, right? What are some of the kind of most promising models that you think are going to give the biggest bang for their buck, or you see as having like the biggest impact on some, understanding pharmacology or basic biology?
There might be too many to count, but
let's go top three.
Yeah, I can at least maybe I'll try to broadly classify or something like that. So for instance, many people think of your body as a series of barriers or tubes, right? So it's like, something goes in this tube and then it has to translate that barrier. So for instance, if you were to give orally delivered drugs, right? It has to go through the gut system, through the GI, and then usually somewhere in the intestine it has to get absorbed somewhere, and there's that barrier there, like an intestinal barrier, for instance. And so there are many models now, like endothelial barriers in the vessels of your blood, right? An epithelial barrier for your intestine, right? And so many of these barriers exist. And I think that many organoid types are epithelial in nature, meaning you can recreate the barrier and then ask questions about transit of molecules across that barrier. And so that's a really promising application, but on many different fronts for many different tissues in your body. Kidney is just a series of tubes to clean your blood. So what does each tube move in and out, right? So all of that can be modeled. And then I think the other thing is disease modeling, right? So you can take the organoids, and especially organoids with known genetic, mutations that caused a particular disease. Like we're even looking at many of them. So you, for instance, looking at autism, like all the autism genes that were changed, and you can get these cells from either the patients or you can create the mutations yourself in the line. Then you can generate organoids out of them and ask, is the electrical activity different, right, in this versus a normal healthy one? And then you can understand those disease states better, and then you can find new druggable pathways within, those organoid systems. Another large class is, I guess, biomarker development. So you can look at, if I gave a drug, did it have on-action activity? And then was there a secreted marker that came out of the milieu that you can then measure right out of the media to verify that had an intended effect?
That's really cool. So what do you think are some of the things that really kind of helped drive the innovation in this field?
So the propagation and culture of stem cells in general, and the knowledge, I know famously, Hans Clevers created one of the first gut organoids back in the day, where it comes not just from a primary embryonic or induced pluripotent stem cell, but comes from tissue-derived stem cells, right? So each tissue often has its own, stem cell progenitor that you can possibly isolate and then use that instead of going all the way back to, a single source of induced pluripotent or embryonic stem cells. So the knowledge of that and then the knowledge of how to culture that has really propagated across so many organ systems now, the ability to recreate many, many of them in depth in a way that we couldn't before.
So you mentioned Hans Clevers. Is that the first organoid that hit the scene, or what's the back history there?
I guess I can't answer to whether it's the absolute first, but I would answer it's one of the most famous for sure, if not the first. And we can go back and fact check me on that later. But essentially, yes, he was able to isolate part of the niche of the gut that's called a crypt. And inside there, it houses the stem cells of the adult stem cells of that epithelial gut tissue. And he determined that they were, for instance, the LGR5 positive population. If you propagated them and then put them into a 3D assembly, they would grow into these sphere-looking, they're almost cyst-looking structures. And that when given certain cues, they would differentiate into like enterocytes of the gut and many other types of cells that are lining the villus, for instance. And so that really became one of the first impetuses of really everyone getting excited about, wow, we can model, and then these cells are real and they really give you the diversity of the human gut. Like, whoa, that's pretty exciting to a lot of people.
Danielle, what do you think the ideal model system is?
I have a very strong opinion about that. I think the ideal system has the pieces that you need to test involved in it. And that sounds like a smart aleck response. But in reality, sometimes when we think about in vitro systems, we can make them way more complicated and it makes it harder to actually answer our questions. Or we don't factor into all of the other pieces of downstream biology that are relevant. So the best model system has what you need to test included in it.
So what you're trying to tell me, Danielle, is that complicated is not always better?
So our listeners can't see me winking, but that was a hard wink. Can you maybe go a little bit further into some of the technological advances? Like, I know, not because I'm a stalker, but I know that your group's research interests lie on also how to do some of these kind of experiments at scale and incorporate like 3D printing. Like, what's that whole shtick? Because that's like a very cool engineering aspect.
Organoids can also just be original starting material that you put in a more complex framework in 3 dimensions. So, for instance, organs on chip, and those words are defined by, let's say you imagine a small microfluidic device, maybe about the size of a thumb drive. And you'd have some input, say media coming in and output media is going out. And inside of that thumb drive, you have a very complex, be it printed or molded system where you would maybe place the organoids amongst, say, some vasculature or things like that to make it much more a realistic environment for what they would see in the body.
Is that like, you'd mentioned that a lot of times when people think about the body and try to break it down, it's like systems of tubes or whatever. Yeah. So are you putting in the barriers that would be there biologically?
Yes, exactly. And I always come at things from an engineering standpoint because that's kind of my primary training. Oh. So I just think of the body as a series of tubes that, does different things. And some, you have pulsatile flow in some regions, you have laminar flow in other regions, and using engineering techniques like that have been miniaturized, right? You can simulate all these things, simulate the pumping of blood, simulate the shear stress on the endothelium of your blood vessels that feel that. You can simulate actually the breathing motion of the lung. That was one of the first organ-on-chip systems. So I think that this dynamicness to how we culture the cells and the engineering going behind that is another major technological advance. And what it does is, I think I had mentioned that there's sometimes some immaturity to the organoids. Mm-hmm. And when you put them in these systems that apply forces that the body always has, mm-hmm. so cartilage can get compressed or, the endothelium can feel the shear like blood going by. It actually forces increased biological maturity that much more mirrors what is actually in your body. So now you can kind of take the organoids to a whole nother level by placing them them in the physiological context in vitro that simulates the body better.
All right, so tell me about this 3D printing, because in my mind I think about some pen or some special printer, but how is this happening in the organoid space?
Yeah, so 3D printing is a technology that's now, burst on the scene, and people even have 3D printers in their homes, and, you can use them to make little cool, like, Yoda statues and stuff like that. It can also be used, if you formulate it correctly, right, to actually print living cells. You can also print, so what I want you to imagine now is, squeezing through a small nozzle, a needle nozzle, right? Some sort of gel that holds these cells in place, and that you can make shapes with, right? And sometimes it can be very small, and sometimes they can be larger assemblies. So if you were to do organoids, and the organoids are fairly large already as a starting material, this would be something you could see with your eye, like printing them in a certain assembly. So what groups are really working on now is how to kind of specifically place the organoids to what I call it nudging biology. Mm-hmm. So meaning like you just try to give the right starting conditions, and you hope that the biology does the work on the micro scale for you. So for instance, you want an organoid to be vascularized, maybe you just place it in a gel and then you place the progenitor cells around it, and you could print that, or you can kind of like hand, pipette that around it. And then you kind of nurture that system with the right kind of feeding and care. And then eventually maybe you'd get a perfusible vascular network, which the organoids are sitting in and around in 3 dimensions in a printed chip. And then you could perfuse that whole thing. And then you, with the cues of shear and the right nutrients, maybe you even create a gradient across that chip. And then you kind of nudge the organoid biology to kind of mature.
Right. So it's like suggested placement.
Yeah.
Okay, so we've talked so much about the benefits, and I'm going to ask you big picture, but I also want to ask you realistically, like, where do you see the biggest limitations now, and where do you think we should be, busting to, deal with it? I don't know.
Yeah, there are quite a few. As much as I am an advocate, I'm also a realist, and also, in my everyday work, I really apply the scrutiny of, can this model really stand up to the question that I'm asking? And really, there's a couple of well-known problems in the field. One is that organoids are known to typically be fairly immature if they're derived from induced pluripotent stem cells or embryonic, because it takes you all the way back to like a, super early state in the womb, and then you've got to kind of mature out of that state for a really long time, like I was giving you the example with the brain organoids earlier.
It's a lot of steps to artificially coordinate.
It's a lot of steps, right? And luckily, biology actually gives it to you for free in many cases. So you do the first couple of steps, and then a lot of times after those directed steps, the cells do it to each other, because there's enough cell types in there that can do a lot of talking to each other through secreted cytokines, and then those drive the changes from there. So you actually can just stop worrying about it, and you just keep changing their media, and they just do the thing, which is super cool. But the problem, there's a couple of problems. One is the immaturity. So when you get to those stages, you ask, okay, so I have a 6-month-old brain organoid. Is that going to be like my 90-year-old Alzheimer's patient?
Probably check, trust and verify.
might want to check that. So these are some of the challenges. Also, many people have realized that when you derive these, they don't come with an innate immune compartment, for instance. They don't also come necessarily vascularized. So there's some things that are missing. So they're going through this development process, and even when you look at histology of these tissues, you're like, wow, this is like, even the architecture is somewhat replicated. But almost all of the systems, the vasculature might, if you're lucky enough, maybe it forms, but then it kind of dies with time. It's not perfused. You don't have a heart pumping it, right? And then that leads to, one, they're not homogeneous. Each organoid does not look like a carbon copy of the next one. Mm-hmm. And they're missing these key elements, right? Immune system integration, vascularization. And then in some cases where organs have to stretch and do things like the lung, like they miss those cues too,
like
right? Mechanical cues. And so they're imperfect representations of the human organs, but they're the best ones we've ever had in a dish.
Yeah.
If that
Yeah, and I feel like also maybe it's not necessarily a down point. It just depends on what question are you trying to ask. Like,
right.
what's the appropriate for the appropriate question?
Yeah. Always a thing.
So how do you deal with the inconsistencies that are there so that you still get meaningful data?
So you do have to create a lot, right? And they are scalable. So you can put them in, every well of a 96 well plate, or people are working on 384 wells now and culturing them. And so you can start just to get a numbers game. You can produce a lot of numbers. A lot of times there are pre-QC steps that also happen, so QC standing for quality control, that happen on the organoids with time. So you can look at their growth rate, and you can use that as a measure of quality. You can kind of separate some of them out and just, look at them. Are they growing well, or is this like historically how they have grown? And it is well known that different donors grow at different rates. Different donors would create slightly different populations. And so researchers have started to take this into account and use, for instance, design of experiments across a plate to determine which lines need which concentration of which thing, because it's not always the same. So they have gotten to that level of detail to really,
amazing.
to really make it consistent enough to use or to trust.
Does that also mean you can simulate more like population variability to kind of, how do I want to say this? Like, I feel like kind of like how you're talking about, traditional cell culture, it's like very much in a vacuum. But like we talk about like health equity and making sure that we're designing and testing our therapies, not just for one person, but for all the different people that are out there and different biological backgrounds and diversity. That seems like it's also another place where you can really introduce that in a way that some preclinical models and animals might be lacking.
Yeah, my goodness, I have so many answers for that. So this is one of like my passion projects. So modeling human diversity is as itself a whole field. Now, I'll first describe how you could apply the technologies that I was just talking about with Organ-on-Chip directly to that question. So for instance, in your body, like say in your kidney, there are some people that are born with 100 or 1 million nephrons per kidney is usually what people have. And a nephron is one singular unit through which a blood goes, gets cleaned, and then the liquids get cleaned, the water goes back, and it goes to urine, okay? So there's 1 million of those in most people's kidneys. Some people have 200,000. But they have to still clean the same amount of blood in the same amount of time, right?
Yeah.
So that means they have a completely different flow rate, right, in those nephrons than the people that have 1 million. And so even with the same cells, you could use engineering to then show that there's actually differences that the cells feel directly. One having like a hyperperfused situation and the other having, I guess, what would you say, normal, right? And there is pathological, considerations to the hyperperfused situation. So that's one way that just technology alone, you could bear that out. Another way you could look at things is say, okay, now I want to create organoids from many different individuals, and then I'm going to test them separately, right? And I'm going to say, oh, I found that this gut organoid from this person has a different metabolic regulator than this one, and therefore they behave differently to these drugs. Now you can start thinking about patient stratification and those types of strategies. Or you can say, oh, I want to be more inclusive in my research, in my preclinical research. So now I'm going to make sure I have the same amount of male and female donors on my dish when I test. I can actually be more preclinically inclusive. And then the third way you could do it is some really unique approaches people are trying now, which is called like village in a dish approaches, where you actually start with, many different iPSC lines or many different starting cells from many different people, and you put them all in the same ball, like in a dish. Oh. And then you give them factors, and then you try to figure out what cells fell out of that mix. For instance, what would be either more toxic or more regenerative to a certain population. And there's a lot to work out there, but that limits the amount of scale you have to do, you know what I mean, to get that same question. I think this field is really nascent in this area, still really working on this, and there's a lot of kinks to work out. But that would kind of mean you wouldn't have to do every single test per patient and have enough automation to make that work.
Yeah, it's like it also opens up like a way to reduce the burden on the patients that you would take into clinical trial as well, right?
Right.
Yeah, that's super cool.
Yeah.
That's super cool. How has AI and machine learning impacted either, setup for these systems, data interpretation? Like, where do you see it playing the biggest role currently?
Yeah, so one of the richest places that you can get data from these systems is just to capture images. And you can do it in life, right? And you can do it over time, all the time. And so using AI/ML to really analyze those images and get way more out of it than we did before. So for instance, some organoids, they're not all perfect circles. Some of them will actually start to bud and create little, actually there's ones I was looking up this weekend, they create hair follicles, little.
Oh, no.
Yeah, so little. So all of those we call phenotypic characteristics with imaging can be modeled over time and looked at over time. And then if you want to get even a level deeper, if you were to do imaging, in a systematic way and then have an understanding of which cell types look a certain way, then you can start guessing the cell types just off of the bright field images that you're taking.
That's a super exciting area. Yeah. I'm really into that.
Yeah. So that involves a lot of AI/ML and predictions and having ground truth set up and taking thousands and thousands of images of what, a stained cell looks like against its bright field, right? But once you have that down, which, experts around us do, we're starting to kind of integrate those approaches, which would be super rich. It's almost like having a single cell, kind of down level of understanding of where your organoid is changing or shifting.
Are there certain modalities that you think are going to be utilizing these systems more than others? Like, we have traditional biologics that I think a lot of us have worked on for a while, but what about like these cell therapies or nucleic acid-based medicines or even cancer vaccines? Like, do you see any trends in the field for these modalities utilizing it?
All. I
Cool.
think all.
No pressure.
No pressure, right? It's because now people are creating, immune organoids, things that model, like you can take a tonsil and chop up, the tonsil and then put it back into these 3D assemblies and then ask questions about, what kind of immune responses does that model? Or you can even take PBMCs and do the same thing, and from other lymph organs as well. And so the vaccine question that you're asking and what is our immune response generally to drugs, right? Like, could we deal with cytokine release syndrome better, right? When we look at things in a dish, can we predict that better before it goes in the patient populations, which all these immune therapies typically have as a side effect?
Yeah.
So there's kind of like class effects of drugs, like you were mentioning the nucleic acid ones, and those, have been very prevalent. I mean, especially really excited about some of those in the brain because of their potential to transit some of those barriers and get to the right location. And then with cell therapies, for instance, you can imagine creating a system where maybe you integrate your cell therapy into that system and then see if it had a regenerative effect, so you can look at efficacy, using these models. So yeah, I see so many plays across modality for the technology, organoid and organ on chip.
Danielle, I was really intrigued when Kim was talking about Village in a Dish. How does that work, and what can you get out of it?
I mean, I don't really know all the ins and outs of it. I assume it's just to try to kind of increase the biological diversity in a single dish. It kind of trips me out because I'm thinking about it from like a cell biology perspective. Like, I don't even know how you would culture that effectively without having cells like compete for each other with nutrients or what have you. And I would probably put money on this as a place where machine learning and AI would come in with like special biomarkers to sort that out. If we fast forward 25 years from now, like, what do you think is going to be like the best case scenario for where these technologies reach?
Well, I do think that we've been working really hard on changing the kind of cultural shifts that we have to do. And I would just love to see a future where if you're a preclinical researcher on the program, your first question is, what did the human model tell me? Right?
Yeah.
And then you go to that first, because then we've built years of trust, just like we have years of trust now on the other model. The other kind of hypothetical question I always rumbles in my head is, if we never used animal models, like, what standard would we put these human models under?
Yeah. But you know, that's the thing, like, I can't wait for the time when there's the ability to do more retrospective analysis of, like, these are all the molecules that went into clinical trials and the ones that succeeded or didn't succeed, and then go back and pour through all of that data. Because I think that's where you get the coolest insight into application.
Yeah, and I would say we are already doing some of that, but we could do more. And one area that I would love to see us in the next, you said 20, 25 years is as the clinical trials are ongoing, we should be testing that human's cells in a dish with that same thing and asking how predictive was the dish to the human directly, right? 1-to-1. And then we would be building our confidence not just of general predictions, but of specific human predictions. And we would know much more the limitations of those predictions and where they'd be clinically advantageous to do.
I really appreciate the cultural shift comment because that's something that we don't always talk about, but it's a thing.
Yeah.
So for those early and career scientists out there, why should they drop everything and run to your lab? Because I mean, they'll have to push me out of the way, but
no one has to push. I think the cool thing is that this can augment our current process for quite some time and add so much more rich information. But I think, the hope for all of us is, I don't know the latest statistic, I think it's about 9.6% of medicines that enter clinical trials actually make it, at the end of the day. Mm-hmm. If we moved that number by 1% with this technology, groundbreaking, done, right? Like, that would be, literally you think of 1% as not very much, but in this case, lives and people and getting medicines to patients, I get goosebumps thinking about it.
So when you put it that way, how can we not? But let me ask you, how do we go from creating so many organoids to creating them in a way that we use them effectively to answer questions for human health?
There's been a lot of attention in this field, right? And every other day you have a new organoid type come out or a new paper on how to integrate it in an organ-on-chip device, which I think is super exciting. And all of these proof of concepts need to be there. For us to really use it in drug development, what we need is also the deep study of how these models are benchmarked. So for instance, it is awesome to create a new organoid and it absolutely needs to be a Nature paper. But after that, we need to take the 100 drugs that modulate different pathways and run them against that model and prove to ourselves that it's predictive of human outcomes. And this piece, the connection of the dots between how you gain confidence in our current system and have that culture shift really happen, is data-driven. And we need to generate that data. We need to do it together, and we need to see it as just as impactful as creating a new version of a model. And I would love, and that's what our group really works on, is how to really put these models into absolute use right now to help patients today. And to do that, I would love to see the field really broaden the benchmarking exercises for how these are qualified.
Please sign me up. I'm happy to work with you guys to do some benchmarking.
Okay.
That was a serious thing, though. Okay. Thank you so very much. I mean, this is definitely this has been a great experience for me. I'm so happy to meet with you and have the chance to hear from you. Thank you so much for joining.
Likewise. Thanks so much for having me. It was lovely.
Cool episode, Danielle. There are a lot of takeaways from this, and I finally know what organoids are. A lot of times we're talking about things that seem, a little bit more speculative and a little bit more science fiction theoretically, but this feels really close. Is this actually happening?
This is happening yesterday. It's happening now. And the thing that makes it not science fiction is that there's so much thought into how these systems are even set up and what are the questions that need to be asked by it, that's what takes it out of this theoretical space. It's such a great tool in today's work.
Danielle, you were mentioning the role of AI and machine learning being useful for analyzing maybe the village in a dish model. How do you see that evolving for these complex in vitro systems?
I think it's not just analyzing the data that's generated, it's also figuring out what needs to be in the system as well. Because I feel like AI and machine learning can also kind of help pick out diverse biomarkers and make sure that our system is actually capturing what that village should be in the dish. And I imagine that there's a lot of other cool ways where we can try to use AI and machine learning to make predictions. I mean, there's just so many, any angle you look at it, there's some way that you can use AI and machine learning to improve it.
The possibilities are endless.
Oh, it's true. And that's our show. Thanks so much for listening. If you haven't already, rate our podcast wherever you listen. It'll help new people find us. And make sure to subscribe. If you have any questions about the show, you can contact us at podcast@gene.com. And now for me, it's back to stocking cells.